Hello students, today we are going to study about isolation mechanisms, the first chapter from evolution, paper 303. So what is a speciation? Speciation is one of the most important events in evolution. It is because of the speciation events there are varieties, differences in the life forms which we commonly group them to be called as species. Therefore, in a simple definition, speciation is an evolutionary process by which biological populations evolve to be distinct species, or even the formation of a new species from the parental species is also called speciation or the origin of species. Just imagine, without this event, in the world there would be only one type of a flower, one type of a butterfly, one type of a tree. The whole world would look so dull and grey, isn't it? This word, speciation, was first coined by the biologist named Orator Fuller Crook in 1906. So, what is a species? Before that, we should understand, according to the modern concept, evolution is a mere change in the gene frequencies in the organism of population through time. Now, accumulation of these changes in gene frequencies in a gene pool of a population makes it reproductively isolated from other sister populations and leads to the formation of a new species. To simplify the concept of species, a number of types of concepts have been put forward by different workers. Altogether, almost 21 different types of concepts are available to define the term species. However, the biological species concept non-arguably is the most accepted concept in defining the term species. According to Mayer, the proponent of biological species concept, species is a group of organisms that interbreed only under natural conditions and that are reproductively isolated from such other groups. So, we negate any species or hybrid as a result of artificial insemination or artificial pollination. Henceforth, we group a species which are again reproductively isolated from such other groups, which means it is free to interbreed only within the group of same species. And therefore, there are some mechanisms which prohibit the interbreeding among different groups, thereby maintaining the heredity, the integrity, which are called isolating mechanisms, sometimes also called as isolation barriers or reproductive barriers, because they prevent the gene flow between the biological species. The term isolation mechanisms was coined by Dobzhansky in his work Genetics and the Origin of Species. So, what are the isolation mechanisms? In fact, the proper way to use the word would be reproductive isolation mechanisms that help or are in play to maintain or are maintaining the hereditary integrity among species. There are various ways in typifying the term isolating mechanisms, like Dobzhansky broadly separated the term into geographical isolation and reproductive isolation. The former, that is geographical isolation, as a result of a geographical barrier, or in short may be called as a allopatric speciation event, which we shall study in the more details in the following chapter, and the latter represented as genotypically conditioned differences between species which do not permit the hybrid formation, or in short, the genetic differences among species which result in isolation between mechanisms. Then we have the other way of typifying the mechanism, that is the intrinsic, extrinsic and the intrinsic mechanisms. where extrinsic mechanisms are mentioned above are isolating mechanisms as due to geographical hindrances like distance, mountain, rivers, forest, deserts, ocean, etc. A mountain stands between two similar species, or a river as a result of pressures of evolution, the species evolved differences, henceforth leading to the formation of a new species. The other way of is the intrinsic mechanisms. These are basically the internal features of an organism, or rather the genetic differences among the organisms, which contributes in different anatomical peculiarities, differences, physiological differences, or behavioral differences, which limit the chances to interbreed with another similar species or different species, thereby isolating them. These differences may be translated into usage of different ecological habitats, or say ecological isolation, time or temporal isolation, behavior differences leading to behavioral isolation, etc., which we shall discuss in more detailed way later. The most simplest way to typifying the isolation mechanism would be pre-mating or pre-zygotic isolation mechanisms and post-mating or post-zygotic isolation mechanisms. In short, isolating mechanisms before the zygote formation and after the zygote formation, the details of which we shall study in the coming slides. Coming to the pre-mating or pre-zygotic isolating mechanisms, the first type to be discussed here is the ecological isolation. Ecological isolations is basically seen in those populations or closely related species that occupy different habitats but within the same geographical area. Because they live, feed, and mate in different habitats, they rarely venture out of their habitat, and hardly interbreed. Here in this slide, there are two closely related populations of rana species. One on the top, the red-legged frog, the Rana draytoni, and the bottom one, Rana boyoli, also commonly called as the yellow-legged frog. These frogs have selected habitats, the red-legged breeding in the large ponds and the yellow-legged breeding in the fast-moving streams. Irrespective of being very closely related, of course genetically, they are reproductively isolated from each other due to different habit preferences, where habitat preferences lower their probability of mating. Interestingly, their different habitat selection have resulted in the maintenance of their stable population. It was found that the red-legged frog, here in the slide above, preferred mating with large-sized females compared to the smaller ones. So when in a habitat of Rana draytoni was intruded by another exotic closely related species, Rana catisbiana, or commonly called as a bullfrog. The male red-legged frog made a pair with the large-sized female of bullfrog, thereby losing its mating success. As a result of which, there was a decline in the population of the red-legged frog as because of false pairing and gamete wastage. In temporal isolation, two closely related species occupying the same geographical range or even the same habitat may not be able to pair or find suitable mates as of due to different periods of sexual activity or breeding seasons. The red-legged Rana dateroni breeds from November to late April, whereas the yellow-legged Rana boili breeds from late April to June. Although there may be a small overlap in the breeding time in the same area, the ecological and temporal isolation prevents hybridization. Similarly, closely related fruit flies in Hawaiian, Drosophila persimilis and Drosophila pseudo obscura, the breeding time in the former is late morning and in the late afternoon. There you see, time has got a very important role to play an isolating mechanism. Behavioral isolation, which is also an interesting and important isolating mechanism or isolating barrier in species with complex courtship behaviors, which are usually exhibited by stereotyped call and response signals before male and female pairs. The stereotyped signals are very fixed, and these rituals thus prevent wasted mating effort that would halt gene transmission by inviable and infertile hybrid formation. These behavioral patterns can be seen in the case of New Guinea birds of paradise, which comprises of 42 different species, but each species having its own mating dance which attracts only its own species and prevents a loss of wastage of mating effort. In mechanical isolation, the morphological differences prevent mating or pairing of different species. The example given in the slide is of the snail cell coiling. In some snail species, the cell coiling is controlled by a single gene. So, the left coiling snails cannot mate with the right coiling snails. That is, the synstral cannot pair with the dextral snails. Among animal species with internal fertilization, structural differences of male and female copulatory organs may prevent copulation and deposition of sperm in female genitalia tract. According to Leon Durfer, 1844, genitalia of insects develop a lock and key principle. That is, the male and the female genitalia are so fitted and they are complementary that even a slight deviation in the structure in either renders the copulation, sometimes even leading to injury of genitalia and even death of the species. The diagram shows the various modifications in the structure of genitalia of the three closely related Drosophila species. In another example of mechanical isolation, an interesting example is drawn from the bucket orchid and orchid bee, in which the male orchid bees obtain wax from the bucket orchid, which acts as a strong pheromone to attract the female bees. In the act of obtaining the wax, the bees fall into the bucket holding the wax in the orchid. The route to escape is through a special pore where the flower attaches the pollen sac on the back of the bees and on its way to exit. Now, the bee flies to another flower where the bee deposits the pollen sac to the ovary by the same process. This coagulation is so strong in bucket orchid and orchid bee that if any of the species become rare or extinct, the other would follow. So, the special structures in the bucket orchid and the bee are so complementary that the other species cannot derive the same benefits. Henceforth, a mechanical barrier is formed which results in the mechanical isolation of the species. Uh, Gamete isolation. Till now, what we have discussed about was the mating barriers. However, gametic isolation is a true example of pre-zygotic isolating mechanism. In this case, the sperm and the ova are chemically incompatible and will not lead to zygote formation, where the different species pair and mate but leading to no zygote formation. For example, we have given here is of a sea urchin. Sea urchins synchronously broadcast their male and female gametes, that is, sperm and ova or the eggs, into the ocean, where sperm and ova of the same species may fuse and lead to gamete formation, later developing into a viable larva which will then again metamorphose into an adult. Now, two species of sea urchin, Strongylocentrotus franciscanus, commonly called as the giant red urchin, and Strongylocentrotus purpuratus, which co-inhabit the same rocky intertidal shores of western United States, but they do not interbreed. The reason is the gametes of these very closely related species do not recognize one another as mating species. So the question is, what is not recognized between the two species? The answer to which lies in the differences in the cell surface proteins, because the cell surface proteins determines whether or not sperm can adhere or stick and penetrate inside an egg. Divergence in these proteins can result to gamete isolation. Among species of abelons, which is a kind of gastropods, the sperm proteins lysine dissolves the vitelline membrane of the conspecific eggs. Conspecific meaning the eggs of the same species, enabling the sperm to enter inside the egg. The divergence among these sperm and vitelline proteins due to differences of amino acid sequences which make these proteins inhibit the fusion of gametes, thus leading to gamete isolation. In case of physiological isolation, certain species have established themselves to stop the formation of viable offsprings due to differences in their physiological attributes. For example, in certain species of Drosophila, mating among the members of different species is not successful because the vaginal mucous membrane swells up to few hours after copulation in conspecific species, whereas the swelling lasts for many days in case of heterospecific copulation, as a result of which the eggs, though fertilized in case of heterospecific copulation, perishes inside the parent's body in absence of being laid or deposited outside the parent's body into a suitable substratum, the phenomenon which is sometimes also called as conspecific predominance. So till to this point we had been discussing on the various pre-mating or pre-zygotic isolating mechanisms and barriers, but now we shall discuss on the various post-zygotic isolating mechanisms, that is, isolating mechanisms which operate after the zygote formation takes place. Among the many post-zygotic isolating mechanisms, the first to be discussed here is the hybrid inviability. Inviability, which means hybrids with low survival rate. So in this case, the sperm and ova of two different species may fuse, but the genetic information shared is insufficient, as a result in abnormal development of the zygote or the organism. The embryo dies after few cleavage divisions or sometimes after the birth or even hatching, which results in the low survival rate of these hybrids. For example, let me put forward the example of Drosophila species. Drosophila melanogaster and Drosophila simulans, though superficially and morphologically look alike, they are genetically different. They have incompatible alleles for a nuclear pore proteins. These proteins generally create pore in the nucleus, which allows essentials like RNA, ribosomal proteins, carbohydrates, lipids, etc., to pass through the nucleus to the cytoplasm and vice versa. Now, the difference of these conserved vital gene results to the formation of inviable hybrids. Similarly, another example is the Panthera tigris and leopard. These two sister taxa, the pairing may lead to inviable hybrid, also called as tigers or even as leogers. The zygote of which divide but the embryo miscarries or are stillborn. However, though I put the picture of leopard on the slide, I shall not discuss it here because it fits to the example of hybrid sterility rather than hybrid inviability. Again, an example which I have discussed in ecological isolation, where the two species of rana or frog geographically isolated for thousands of years, but due to introduction of any of the frog into the habitat of another, will lead to incompatible mating between the male red-legged frog and the larger bullfrogs, which results in the zygote formation, which are inviable and thus making a mating vestige. The second type of post-zygotic isolating mechanisms will be hybrid sterility. Here, the zygotes are not inviable, which means it results in the formation of hybrids or hybridization takes place. But the consequence of which is that the hybrid is sterile, which means the hybrids cannot reproduce again. The tigers and lions are sister taxa. However, their mating is usually not seen in the wild because they occupy different habitats. But the mating is seen in some closed areas or confinements, usually the zoos, where they have been reported, of course not in natural conditions, to produce sterile hybrids, though the hybrid possesses viability and are also robust in nature. The picture here is of a liger, the result of the male lion mating or pairing with a female tiger. Another example is of a horse and a donkey. The two species are separated for millions of years, yet people are able to forcefully, or rather are able to pair them unnaturally in forming a mule or a hinny, which of course are both sterile. Again, there are also a few isolated examples of fertile hybrids, like the example of Lonicera fly and American red wolf, which is of course as a result of meeting between the grey wolf and a coyote, and also wheat plants, where they are tetraploid and hexaploid in nature. The last slide to be discussed in post-zygotic isolation is the hybrid breakdown, the example of which is mostly seen in the case of plant species. The two species hybridize and produce a viable and fertile F1 offspring or generation, but the successive generation, that is the F2 and beyond, will lose their viability and fecundity. Thus, they cannot establish themselves as a true population. The example in the slide you see is of a rice plant, where you see both the parents are viable and with high yield. But again, compare it with the F2 you see. The F2 has lost its viability and yield production, which again in successive generations shall lose more of its viability and thus cannot establish themselves as a new species or a true population. Thank you for your patient hearing.